Hello humans on this random planet called Earth, and welcome to my channel. My name is Zelda and I mainly make fashion content, but today I am back with another IB advice video. In this video I am going to give you part 1 of my extremely detailed advice on how to score a 7 on the IB Chemistry IA. And to all of you people out there that got literally zero help on your IA, I absolutely feel you, and I hope that even though this video is super long, it will help you in some way to save time. I will be mentioning some of the same things as in the Bio IA video, but unlike common perception, if there's one thing you take away from this video, it is that not all science IAs have to be the exact same. There are differences between all of them, and I will try and emphasize them in this video. As with the Bio IA, my Chemistry IA was moderated and I ultimately scored 22 out of 24 marks. Unsurprisingly, I lost 1 point for personal engagement and 1 for communications. I know this because I requested the return of materials, which actually broke down where I got which points. For those of you that are interested, this form did not include much except for the aspect that my teachers predicted me 21 out of 24 and not 22 out of 24. And if you're interested, here are my credentials, or more to say, my limited credentials. They're basically the same as for the biology IA and not very interesting, to be honest. I think most of you will also already know this, but here's also the Chemistry IA marking criteria point breakdown, and I will also be linking a table of contents below so you can find the different sections of the video. So now that we have this super long intro out of the way, let's get into the actual video. Okay, so here we have my IA, and I will just be going through it and pointing out different things as we go along. First thing, we have structure. As you can see, I just put a general structure up on the screen, and this is basically the same as the biology IA except for the motivation section, which was included in the background information on the Bio IA, and also the Chemistry IA has a sample calculation that has to be placed prior to the process data, and I will explain the reason for that later. Next, we have some other general notes that you might want to keep in mind. So firstly, you should not have any names, candidate codes, or other identifying factors on your final IA. You also do not need the date you wrote the IA, and you should always include page numbers if you want to gain full communications marks. Starting with the title, this should generally be a more concise and general version of your research question that encapsulates the main aim of your research as a whole. Thereafter, I have my research question, which was, how does varying temperature affect the rate of reaction of the decomposition of hydrogen peroxide to oxygen and water, catalyzed by potato catalase measured by time taken to evolve 10 plus or minus 0.5 centimeters cubed of oxygen gas. Your research question should be a statement in the form of what is the effect of X on Y, where X is your independent variable and Y is your dependent variable. In almost all cases, you should only have one independent variable, and both your independent and dependent variable have to be quantitative. In some cases, you will have multiple independent variables, but this is relatively rare, and usually if you have multiple independent variables, you're doing multiple separate experiments, usually related to the rate of reaction unit, meaning that you still only have one independent variable for each separate experiment. As the IB pointed out, compared to previous sessions in both English and Spanish, there were less investigations that involved more than one independent variable, which is often an unnecessary complication. So really make your life easy and only stick to one independent variable. Either way, you must include the units for both your independent and dependent variables. An important thing to point out is that if you use continuous data, you always have to mention your entire range, so this would include things such as concentration and varying temperature, as well as their uncertainties. The research question should also include how you measured your dependent variable and the uncertainty for that method. In my case, this was measured by time taken to evolve oxygen gas, and I indicated the uncertainty for both of these metrics. In other cases, you might measure something with a ruler, where you would have to indicate the uncertainty of the ruler, or you might measure the change in concentration, in which case you need to indicate the uncertainty of a data logger perhaps. I'm not certain if you have to include the chemical formula of any type of chemical you use, but because both my teachers were really adamant about me including it, I did ultimately include the chemical formula of catalase in the research question. Honestly, I wouldn't do this in most cases because it could potentially contribute to decreased communications marks, but either way, I don't think it will make a huge difference whether you include it or not. Finally, you should include the genus and species name if you used any organism. I should mention that I did indeed make a mistake here by italicizing both the genus and species name. However, since this is chemistry, I kind of doubt that it had a major impact on me losing the points I did. But to be factually correct, you have to include a small case letter for the species name, and it ought to be in italics. Following this, I have my motivation section. Commonly considered the personal engagement section, this is just a short summary about why I chose my topic. This should not be longer than 2 to 3 sentences and should definitely not be titled personal engagement, as the IB absolutely hates this, and long rambles about why you chose a topic as stated multiple times in their annual subject reports. Personal engagement is a criteria that is demonstrated throughout your lab report, and this is only a minuscule part of how to demonstrate personal engagement. As the IB put it, the commonest limitation to achievement in the first aspect of the descriptor was where students failed to show genuine curiosity by presenting a very undemanding research question, where the outcome was too self-evident. Where students presented a research question that reflected a question that they genuinely appeared interested in answering and couldn't already be expected to know the answer, then credit was given easily. They also mentioned the second part of the descriptor regarding personal input and initiative is evident across the whole report. Though they did not specify a list of things that contribute to personal engagement criterion in chemistry, they have presented such a list for biology, and if you pause the video now, you can read through it. While I want to emphasize that there are differences in biology and chemistry, this list should give you a good overview of the things that may be important to consider for chemistry and gaining full personal engagement marks as well. As aforementioned, I lost one of my IA marks here, which is completely unsurprising considering that my topic is one of the most overdone experiments in both biology and chemistry, as it is an extremely accessible experiment for schools that lack equipment. Nevertheless, for the second part of the criterion, I most likely gained the mark because I had a lot of data and considered several other aspects related to personal engagement. Just be aware that if you do an overdone topic like I did, you will definitely lose 1 mark for basically not being creative. This doesn't mean that you can't still score a 7 though. Next we have the background information. This is pretty much a detailed description of anything linked to your research question and information required to understand the reaction under investigation. The way you should organize this definitely depends on the flow of your research, but you always want to separate these into shorter paragraphs to maintain clarity. It can be beneficial for you to just title them in smaller subsections like I did. Here you can see I divided this into just a paragraph directly after the title background information, reaction under study, and finally mechanism of reaction. In the paragraph directly after the background information, I basically just talked a little bit about what hydrogen peroxide is, what exactly it does, and how it is used in organisms, and just some basic information about hydrogen peroxide as a whole. I also made sure to include the chemical formula of hydrogen peroxide because this is chemistry and you should definitely mention that. In the following subsection, I then went on to propose the basic decomposition reaction of hydrogen peroxide as a chemical equation. Note that this did not include the aspect that I was also using catalase. Then I linked my research question to the IB chemistry syllabus by talking about collision theory and the different factors that affect it. This part is really important because you should be linking whatever you are researching to the syllabus. In many cases, it won't be an extremely straightforward theory such as collision theory, but for example, if you're researching colored complexes, you will want to talk about the way that light interacts with electrons and tie it in that way. Just make sure that you definitely tie whatever you're researching into some part of the IB syllabus. Since I was using catalase, which is a catalyst, I discussed the impact of catalyst in more detail than the other aspects of collision theory, and therefore also included the graphs of the decrease in activation energy. Keep in mind that for the IA, you're also graded on how relevant the information is, and in my case, catalysts are a relevant thing to discuss. Whereas if you're investigating temperature, you might only want to mention it on the side and then focus on the effect of temperature. You really need to adjust this type of information depending on what your report is about. From here, I transitioned into a basic discussion about catalase specifically as an enzyme and what exactly it does, and thereafter I discussed in a lot of detail how the structure of catalase is involved in the reaction under investigation, which is why I also included an image of the structure of catalase and the porphyrin ring located at the center of catalase. I really want to emphasize how important it is to thoroughly discuss what exactly is going on at the chemical level and focus on your specific reaction rather than just the broad concept explored in the IB syllabus. In the past, the IB has commented that a lot of students do not go into enough detail about their specific reaction and only broadly discuss it. From the IAs that I have reviewed so far, this is definitely one of the biggest problems I've noticed as well, and I would really recommend that you focus especially on this. Many students tend to sort of scratch the surface of random things related to their research question, but they don't go into depth about any of the things they talk about. As the IB stated, background information showed a wide variety of relevance and depth. Many candidates clearly focused on systems under study and provided relevant equations and even structures when pertinent. In other cases, background information was general rather than specific, with lengthy sections including such material as health concerns, totally unrelated to introduction and research questions. By the way, I would like to highlight that they have a bunch of grammar errors in these reports, making it kind of difficult to read. Anyways, for example, if we consider the same experiment but for biology, it would indeed likely be enough to simply talk about the basics of what exactly catalase and hydrogen peroxide are, and the aspect that catalase expedites the decomposition of hydrogen peroxide by lowering the activation energy with a short mention of collision theory. However, for chemistry, you should be talking about things such as multi-step reactions, and what exactly is being oxidized or reduced, and where within each of the compounds this occurs. In my investigation, the reaction occurred on the porphyrin ring, which is why I mentioned it. I'm fairly certain the lack of detail is one of the things that causes a lot of students to lose a large number of marks that they really don't have to. So just avoid that and include a lot of detail, guys, and make sure it's relevant. In most cases, your background information should be about 1 to 1.5 pages. An easy way to check if you have mentioned the bare basics that need to be in your background information is by looking at your research question and seeing if you have written an entire paragraph about any chemical compounds and organisms that you are using in your reaction. The background information section will likely have the most sources in your entire lab because it should be very detailed in case you have not gotten the memo yet. This is definitely the part of the IA where you want to be extremely careful about plagiarism, so really cite anything you take from somewhere else. Moving on, we come to the choice of materials section. While officially this isn't a requirement, it aids in maintaining a good structure and is a useful section to demonstrate personal engagement. It has also been stated in subject reports that many students benefit from using such sections. Here you want to include a description of how you came up with your experimental design and why you chose the materials that you did. For many chemistry students, this includes an explanation about why you had to modify your research question, since many schools have very limited scientific equipment, especially for chemistry, and therefore a lot of us aren't able to conduct experiments that are particularly challenging. This was also the case for me. So for example, though I initially wanted to calculate the activation energy required for my reaction, this was not possible because my school was unable to provide the resources for this investigation, even though it already required relatively basic equipment, and therefore I mentioned that. If your school likewise does not have extremely expensive equipment available and it limits the scope of your research, this is definitely relevant to mention as it indicates that you're not just being lazy and picking easy topics to avoid challenging work. If you experimented with different chemicals for your reaction or catalyst, you also want to mention why you settled on the catalyst or reactants that you ended up choosing. For example, I experimented with mangan 4 oxide and iron 3 oxide, but ended up choosing catalase. Because during my preliminary experiment, mangan 4 oxide ran out, and also it was quite difficult to work with because it kept sinking to the bottom of the test tube. If you work with a data logger, you also want to mention why you chose that specific type of data logger. For example, did you choose a pH data logger or a pressure probe, and why? Then if you're working with concentrations or other types of continuous data, you want to explain why you limited your investigation to that specific range. For instance, I explained that I limited the concentration of hydrogen peroxide and catalase based on my preliminary experiment and the aspect that the reaction occurred too fast with a higher concentration of catalase and too slow with a lower concentration of catalase. Note that to gain full marks, you have to justify why you specifically chose your independent variable and dependent variable, and this is a great section to do so as well. In many cases, the justification for why you chose your independent variable is because previous research has indicated the specific range of concentrations or temperatures, etc. is most effective to investigate, and you lacked higher quality equipment, so your chosen independent variable was simply the easiest to control, while the dependent variable is most likely the easiest to quantitatively monitor. Lastly, you want to explain why you chose the apparatus that you did. Specifically, if you are measuring volumes of liquids, you want to be incredibly careful, since most students mess this up and use the wrong equipment to measure it. You cannot use beakers to measure volumes, and instead you should opt for things such as volumetric flasks and graduated pipettes. Also note that this becomes especially complicated if you're working with hydrated compounds, and you should be very careful with that. If you're interested, here's a list that the IB published of common mistakes that students made. I recommend you pause the video and read through this, as these are all things that could be discussed in your choice of materials section. Next, we explain your hypotheses. This is relatively self-explanatory, but a few things to point out are that both your IV and DV have to be included in the hypothesis. In my case, the IV was increase in temperature, and my dependent variable was the time taken, which was subsequently used to calculate the rate of reaction. You also have to include the range of your dependent variable if you are using continuous data, such as things like increasing concentrations, temperatures, etc. And you have to include the units and uncertainty of your measured dependent and independent variables. Also include how you measured your independent variable and dependent variable. And finally, your H1 should probably also be backed up by a scientific source, meaning a published and credible research paper. Moving on to variables. You start by naming your independent variable again, include the range if relevant and the uncertainty, and then you explain your dependent variable and how it is measured, as well as a short description of what equipment you use to measure it. The final variables are your control variables, and these should always be set up in a table. The table should look something like this, where you have what the control variable is, the reason for controlling that variable, and the method of controlling said variable. As an example, we have the concentration of catalase. The reason for controlling this was due to biological variation of S. tuberosum. Different ones may contain different concentrations of catalase and other compounds, which could alter the chemical reaction and concentration of catalase, and thus the measured rate of reaction. A single stock solution ensures that the concentration of catalase and other compounds is the same for all trials. My method for controlling was then blending all potatoes together to form a single catalase stock solution that is mixed thoroughly. Note that human rate of reaction will never ever be a control variable, because even if you do use the same person to measure the time taken every time, there is an inherent flaw in the rate of reaction of a human, and therefore they will never be perfectly precise, and hence it cannot be used to control the accuracy or reliability of any data gathered. So make sure you don't use that. Likewise, your classroom atmosphere or environment cannot simply be said to be a control variable, and be aware that this is a mistake that many students make in chemistry, especially when it comes to controlling temperature. Firstly, you must specify what specifically about the environment you are referring to. Are we talking about air humidity, air pressure, temperature, or something else? And also, you have to explain exactly how you controlled it. Staying in the same room the entire time is not a method of control, seeing as airflow can change, altering the temperature or humidity, and various other things can happen. If you assume any of these things to be true, this will be a big problem, particularly for your calculations. Many calculations have an input factor such as temperature or pressure, and your final calculated results will be altered significantly if you do not control it, meaning you'll most likely lose a lot of marks for analysis and evaluation, so please be super careful with this. A few things the IB has mentioned that are reasons for the methodology to be considered inappropriate were failure to measure the relevant dependent variable, failure to collect data related to sufficient values of the independent variable, and failure to control or at least monitor relevant control variables that clearly would impact the finding. This was the most common of the methodological weaknesses. If you are trying to control surrounding temperature or reaction temperature, you could instead try to conduct the experiment in a water bath, for which you maintain a consistent water temperature. Or you could set up a thermometer in your classroom and continuously monitor the measured air temperature. If you're trying to control pressure, ideally you'd like to use a pressure probe, or alternatively, you could use a barometer, which is somewhat less precise but also functional. If you're super desperate, what you can do is find a local weather report that reports pressure on an hourly basis, which is what I did since my school did not even have a barometer. But be aware that this raises a lot of issues in terms of precision and accuracy, because it's not measuring the pressure specifically within whatever area you're experimenting, and the pressure can differ even between inside a room and the outside environment, which is usually where weather forecasts measure things such as pressure. In total, I believe you need 6 variables. I'm not 100% sure about this, so please do double check that. But either way, you want to name at least 2 variables more than what you're required. You do this because even if the IB rejects 2 control variables, you still have the minimum number of required control variables, and consequently, you will not lose any marks. In contrast, if you are required to give 6 control variables and you present 6, there is a high probability that at least one of these variables will not be considered a proper control variable, and so you will easily lose a mark there. On to materials and apparatus. This is quite self-explanatory, but this is just a list of materials and apparatus you used, as well as the quantity or volume you used. Make sure you are consistent in terms of the order, and ideally you also want to name the brand of the equipment or source of material you used. You also once again need to include the uncertainties of any equipment used. For example, we can see here I wrote 2 times 25.00 plus or minus 0.06 milliliter brand GmbH graduated pipette. Here the quantity is named first, the volume, and then the uncertainty is named, and finally the brand of equipment and the equipment is named. As far as I know, there is no required order except for that typically the quantity of whatever the material or apparatus is usually named first, but even that I don't think is required. Moving on, we have safety, ethical, and environmental precautions. As the IB stated, safety issues continue to be fairly well addressed, although often instructions were general rather than specific. IB World School laboratories should have adequate fume hoods, and certain reagents or products require their use rather than a simple ventilated area. To gain full marks for this section, you have to include at least one consideration for safety, ethical, and environmental concerns, and explain how you avoided or minimized the impacts of these. For instance, here I said, in consideration of ethical concerns, it was chosen to obtain catalase from plant sources as
opposed to animal sources, such as liver, to ensure no animals were harmed for this experiment. Here we see the ethical consideration and the way in which we minimize the impacts of using this methodology. You basically have to do something like this for each safety, ethical, and environmental consideration. In rare cases, you will not have ethical or environmental considerations. If this is the case for you, which is likely the case if you are running simulations, you simply write that there were no ethical and environmental considerations and explain why that was the case. Next, we have procedure. Here you basically just write in detail what you did. What I mean with in detail here is that you write down literally every single step of what you did. If you dried your test tube with a paper towel, you write that down. If you cut your potato transversely, you write down that you cut your potato transversely and not vertically. If you calibrate the pH meter, you write that down. You kind of get the point here. Also make sure that you list all equipment you used with the specific size if relevant. A simple way you can check if your method is detailed enough is if you go to your materials list and each item named there appears in your method somewhere. According to the IB, there is no preference in terms of writing it in prose or recipe style. Nevertheless, I would recommend that you generally write in recipe style because it often forces you to be more concise, as otherwise the list makes little sense. It may be valuable to separate your procedure into several subsections like I did, depending on your experiment, as this could potentially help you maintain clarity and gain communications marks. Here is also where you again demonstrate safety, ethical, and environmental considerations. For example, in the safety considerations, I mentioned that following the experiment, all materials used were first diluted with water and then poured down the chemistry laboratory disposal drain. In my procedure, I then wrote in step 4.12, the final mixture was diluted with tap water and poured down the laboratory disposal sink. If you dilute a solution, it is likely also effective to present your dilutions in table format after whatever step in your procedure discusses the dilution, since it is simpler to read for the examiner. Also, if you are using some sort of strange method to filter something or anything else along those lines, you might like to add a diagram that further explains your method through annotations. For example, here in figure 6, you can see how I filtered the catalase solution, which likely wasn't a necessary picture, but it still adds to the description of the method, so I think it was acceptable. Make sure that any picture you add, though, is actually relevant and adds information. Following the method, you need a diagram of the experiment. Most commonly, this will be a diagram of your setup or a diagram of how you measured your dependent variable. I don't want to give too much advice here because from what I have read, there seems to be a bit of controversy surrounding what is relevant and what is not. But this image should help further explain your experiment, and therefore it should not be something generic that is simply added for the sake of adding a diagram. Note that adding superfluous diagrams can contribute to the loss of communications marks and potentially also marks in other sections of the grading criteria, as the IB is mentioned in the past subject reports. Just like your tables and other figures in your IA, these images or diagrams also always need a descriptive title. It is not enough to say diagram of experimental setup. A tip one of my biology teachers said is that if you cover up your table or diagram and solely read the title of the figure, you should know exactly what is included in it. You should also give credit to any images you took off the internet, and if it is your own image, you can simply state researcher's own image. I'm not sure if you have to include the date the image or diagram was created, as I've heard conflicting information about this, but I included it. After that, you need your raw data table. The title for this is generally written in the format the effect of x on y, where x, again, is your independent variable and y is your dependent variable. Usually what you want is that your independent variable is placed on the y-axis of your table, in my case the varying temperature, and the dependent variable is placed on the x-axis of your table, in my case the time taken to evolve oxygen gas. You need to include the units of your independent variable and dependent variable and the uncertainties in the header of each column. Do not put these in the cell, as you are guaranteed to lose communications marks for that. For example, you can see the title of my dependent variable, the units, and then the uncertainty, plus or minus 0.5 Kelvin. And then you can also see the uncertainty of my independent variable here. If you measured any control variables, which most likely you should be measuring, you also want to include that raw data in the table. In my case, this included air pressure, which I gathered from a local weather forecast as aforementioned, and then I also mentioned that in the footnote. Convention is also that all numbers should be centralized in tables. Below this, you have your qualitative data. These are basically relevant observations that are not quantitative. For example, if you notice a color change or you realize one of your test tubes had something strange in it, you may want to note that down. The key here is that it should be relevant. If the strange thing in your test tube did not result in your trial becoming an outlier, it may not be worth noting it down. Generally, bullet points are probably the easiest, but I think you could also do it in a little text, but I'm not really sure. Either way, you should not use images in replacement of text for qualitative data, as the IB has discouraged students from doing so in the past. They stated in their subject report that with regard to qualitative observation, it was good that less students used pictures instead of recording their observation. Interpretation of images is subjective, and as already observed in previous reports, this should be discouraged. An incredibly important thing to mention in this section is also any increases in uncertainty, and a short explanation as to why you increase the uncertainty. Note that in my case, my teacher forced me to remove it from this section because he was confused about the section and, to be honest, had no clue about the IA in general. However, the vast majority of my other advisors have recommended to place it here, and to the best of my knowledge, that is also preferred by the IB because it makes logical sense here. Things like human rate of reaction should be increased, and the increase has to be justified. The justification in most cases is fairly easy though, since you just need to say what the flaw in the initial measurement was. For example, you increase the uncertainty of the water bath temperature because you notice small fluctuations in the water bath that exceeded the standard uncertainty of plus or minus 0.01 degrees Celsius, or whatever the issue was. Note that you later have to use these individual uncertainties to derive your uncertainties for the final calculated value.